صباح الخير. ناخد هلا الشابتر 9 دوكيمنت 6 هيدا. اوكي، page 194 and 195. Here what do we have? Objectives. What are the functional synapses? Study of excitatory and inhibitory synapses. Definition, comparative study, and the flow of ions in case of excitatory synapse and inhibitory synapse. What happens at the level of the postsynaptic cell or postsynaptic neuron? That is, how EPSP, excitatory postsynaptic potential, and inhibitory postsynaptic potential, how are they created? And then, what is the fate? What happens after the creation of EPSP and IPSP? EPSPs or IPSPs, or EPSP and IPSP, depending which is found or available at the level of the postsynaptic membrane. What are the kinds of summation? Definition of temporal summation, spatial summation, and tempospatial, which are mixed temporal and spatial summation. And what happens at the level of the nerve center, for example, spinal cord and or brain, of the different or many PSPs received from presynaptic neurons. Here, what's an excitatory synapse here? Case 1. And what is an inhibitory synapse? A synapse is said to be excitatory if, a synapse is said to be excitatory if it allows the nerve message to be transmitted from presynaptic neuron into postsynaptic cell. For example, in neuron, postsynaptic neuron. Here, an EPSP is formed. If the EPSP reaches threshold potential here, an action potential is created, like this. Like this. In case of inhibitory synapse, if the nerve message at the level of the presynaptic neuron will not be allowed, blocked, stopped, will not be allowed at the level of the synapse into the postsynaptic cell, here the postsynaptic cell, neuron or muscle or gland. If it is not allowed, then the synapse is said to be inhibitory. What about the flow of ions at the level of the postsynaptic membrane? In case of excitatory synapse, there is a massive inflow of transport of Na plus from the external medium to the internal medium. Here, the membrane will be less electronegative, and action potential may be created if EPSP reaches threshold potential. However, in case of inhibitory synapse, this leads to, there is an Cl minus inflow or K plus outflow. Yeah, depending on the kind of the neurotransmitter, which could be excitatory neurotransmitter, or here, and here it is inhibitory neurotransmitter. Here, you notice here the Cl minus inflow or entrance, here the membrane or the interior becomes more electronegative. VR minus 70 becomes VR minus 75, minus 80, and IPSP is 4. This is a local depolarization, and this is a hyperpolarization. فإذا تعرفنا على PSP ممكن يكون EPSP، ممكن يكون IPSP. What happens at the level of the excitatory synapse and the inhibitory synapse withdrawing? When it is activated, the Na plus channel will be activated, opens, allowing the inflow of Na plus from the external medium to the internal medium. So Na plus depolarizes the membrane, increasing the likelihood of firing and action potential. However, in case of inhibitory synapse, the binding of the neurotransmitter here the binding of excitatory neurotransmitter, here the binding of the inhibitory neurotransmitter to its receptor. The Cl- channel here, for example, hyperpolarizes the membrane, which it becomes more electronegative. So decreasing the chance of firing and action potential. Why? Because this becomes farther far away from threshold potential. يبعد كتير عن threshold potential. So no possibility of creation of a new message or a new action potential here. So no response here. More and more EPSP and IPSP. This is, and what are the kinds of PSPs? Postsynaptic potential, PSP. EPSP, excitatory, postsynaptic, here S, potential. Provides a small local depolarization, generally results from opening of an channel that manifested by inflow of Na plus ions. This is EPSP. For example, this minus 70 millivolts rest potential here, for example, minus 60. If it reaches threshold potential, then action potential will be created like this. IPSP, inhibitory postsynaptic potential, provides local hyperpolarization. General results from opening of K plus channels, leading to outflow, not inflow, and Cl minus inflow. And this is far away, very far from threshold potential. This is called IPSP. كمان هيدي صورة توضيحية هون للي شفناهم بالأول بس بطريقة أخرى حلوين كتير. هون EPSP here the opening of Na plus channel after binding of the neurotransmitter molecule to its receptor found on the Na plus channel ion or receptor of neurotransmitter, allowing or provoking the opening of Na plus channel so that Na plus ions will be entered or transported to the inside. So here an EPSP will be created. On rest potential minus 70, minus 65 بهذا المثل. As a sodium ions in flow inward is responsible for generation of EPSP. بالمقارنة IPSP, the neurotransmitter here binds to the inhibitory neurotransmitter, binds to its receptor here on Cl minus channel, leading to provoking the opening of this channel and allowing the Cl minus to enter into the inside of the postsynaptic cell and creation of what, or generation of what, of IPSP. Here, what do we have here? We have many synapses. This is excitatory, this is excitatory, and this is inhibitory. Question is, what is the result? What happens here? Do we have a new nervous impulse? Do we have EPSP only? Do we have IPSP? What will happen? Here, when neuron A, which is an excitatory synapse, is stimulated twice. هذا EPSP1 هذا EPSP2 هذا البي stimulated twice. EPSP1 EPSP2 or عندك EPSP1 2 3 4, which reaches here the threshold potential and action potential is created. Okay. The question is now, what are the types of summations? Summation could be temporal, could be spatial. What is temporal? Temp بالفرنسي أو بالإنجليزي Latin English means time. Due to successive stimulation of same neuron. عشان هيك كتبتها بالأحمر. Temp, same neuron. Here, the electric signals are received from presynaptic neuron are separated by time. However, spatial and space, distance, simultaneous stimulation, simultaneous at the same instant, at the same time of many neurons أزرق فيهم in parallel, where the electric signals received from presynaptic neurons are separated by space. Here in neurons, and here we have the same neuron. Successive stimulation, stimulation 1, 2, 3. Here EPSP 1, 2, 3, and may reach threshold potential and action potential is created. Here, stimulated 1, 2, 3, also several EPSPs are formed and action potential may be created. إذا نشوف فرق بين الtemporal والspatial. Stimulations here successive of the same neuron, single neuron. Not many neurons are involved. Here the presynaptic and here the post. However, in the spatial, here these three presynaptic neurons at the synapse S1, S2, S3 synapses, and each one is stimulated. All of them are stimulated at the same instant, at the same moment. Okay. إذا تعرفنا على السباشال والتمبورال. كمان هون صورة توضيحية أكثر. هون no summation. No summation. هذا التمبورال. Successive stimulations of the same neuron. Okay. EPSP1, EPSP2, EPSP3. Spatial. Neurons are in parallel. Presynaptic, 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 and postsynaptic. That are stimulated at the same moment, same instant, but the signals are separated by space and not as in this case. This is spatial. كمان هون صورة توضيحية أكثر للtypes of summation. Steps بتبلشي فيهم 1 2 3. Steps on 1 2 3. كمان حلوين ما صعبين. هذا spatial أزرق وهذا الأحمر temporal. Okay. Here we have three excitatory synapses. Here, here, and here. And one inhibitory synapse. These are presynaptic neurons. And this is also a presynaptic neuron. And this is the postsynaptic neuron. The question is how EPSPs and IPSPs are integrated so that a decision will be made or created so that an action potential can be created or not. This depends on the EPSPs and IPSPs done here at the level of the postsynaptic membrane. Here, if you have E1, E2, E3, etc., here EPSP, here IPSP, and the EPSP, and the IPSP. Here we have of the same neuron, we have three successive simulation: EPSP1, EPSP2, and EPSP3, leading to an action potential because the sum of EPSPs, the algebraic sum of EPSPs reaches or exceeds this threshold potential so that an action potential is created. Also here, and that the spatial summation, what do we have here? We have E2 and E3 on these neurons, EPSP1 and the EPSP. And here, when they are successive, but for different neurons, the resultant EPSP reaches or exceeds threshold potential so that an action potential is created. هون كمان توضيح. These are excitatory synapses here, 1, 2, 3, and this is an inhibitory synapse, and this is the postsynaptic neuron. Can action potential is created? Here, they are integrated, اندماج. This depends on what? Depends on the resultant of the algebraic sum of both EPSPs and IPSP summation. That is the nerve message, for example, the frequency of action potential at the level of the postsynaptic neuron and the postsynaptic neuron is either amplified and frequency increases, attenuated and decreases, cancelled and no response, or stabilized, or the nerve message can be transmitted without any change at the level of the postsynaptic neuron. It transmitted from presynaptic to postsynaptic neuron. فإذا تعرفنا شو هم تعرفنا على overall review. تعرفنا على الفانكشنال synapses. يعني synapses are classified مثل ما ذكرنا سابقا, are classified according to function into excitatory and inhibitory. تعرفنا على ال EPSP and IPSP which are postsynaptic potential. تعرفنا على summation, شفنا شو الفرق بيناتهم temporal and spatial. واخر شيء تعرفنا كيف بيصير في integration of PSPs and what is the result. هيدا كل شيء, وراح وراهم تشوفون هلا support videos. Thank you. Bye. هيدا support videos.
A neuron transmits nerve impulses through itself, but when it's time to send a message to another cell, most neurons will send chemicals to the next neuron across a very small gap between them. The junctions between neurons and the next cell are called synapses. There are three major types of cells that neurons will form synapses with: sensory receptor cells, other neurons, and effector cells. A sensory cell could be part of the eye, ear, nose, skin, or any place where senses are used. This sensory cell is connected to the skin and will pick up pain sensations. The pain message may be sent to another neuron that could be part of the spinal cord or brain. These intermediate neurons are also called interneurons. The message can then be sent to a cell that will have an effect on another cell, so they're called effector cells. In this example, the effector cell is going to signal a muscle cell, which would move this area of skin away from the source of pain. These three cell types together make a simple reflex arc, going from sensation to the central nervous system to an action. The chemicals released into the synapse are called neurotransmitters. Many neurons have a very small fluid-filled gap between the presynaptic cell and the postsynaptic cell. This gap is called the synaptic cleft, and it's only about 20 nanometers wide. It's a really, really tiny gap. Now that we know some of the basics, we can look at the steps of synaptic transmission in more detail. First, a nerve impulse in the presynaptic neuron reaches the end of the membrane. This causes calcium ions to diffuse into the cell through channels in the membrane. The influx of calcium causes vesicles with neurotransmitters in them to move and fuse to the cell membrane. The neurotransmitters are released into the synaptic cleft by exocytosis. They diffuse across the synaptic cleft and bind to receptors on the postsynaptic membrane. The binding of neurotransmitters triggers sodium ion channels to open and pass the threshold potential. The action potential is propagated along the postsynaptic neuron. Then the neurotransmitters are broken down and removed from the synaptic cleft. If there isn't enough neurotransmitter binding to receptors, then there won't be enough sodium flowing into the cell to reach threshold potential. This means that there is an all or nothing response. Either there is enough neurotransmitter to start the action potential and the impulse is sent, or there is no impulse. In this case, there was not enough neurotransmitter released to trigger an action potential. So the neurotransmitters will be broken down and the sodium-potassium pumps will restore resting potential in the postsynaptic cell. There are many different types of neurotransmitter, but we'll look at just one in more detail right now, acetylcholine. This neurotransmitter is made of an acetyl group and a choline, which must be combined together in the presynaptic cell before it's packaged into a vesicle. Then it can be released into the synaptic cleft. After acetylcholine binds with a receptor, it is rapidly broken down by acetylcholinesterase, an enzyme that's present in the synaptic cleft. The acetyl group and choline can then be reabsorbed by the presynaptic neuron and combined again into acetylcholine for reuse. One type of insecticide takes advantage of the acetylcholine receptors to paralyze and kill insects. Neonicotinoids are synthetic compounds that are similar to nicotine but bind permanently to the acetylcholine receptors. The acetylcholinesterase cannot break the neonicotinoids down, and the neurons can no longer receive messages or send action potentials. This is what leads to the paralysis and death of the insects. The effects are very small in humans and mammals, but the neonicotinoids don't discriminate between types of insects. Honeybees and other beneficial insects are inadvertently killed by neonicotinoids too. Thanks for watching this episode of Teacher's Pet.
The postsynaptic potentials that are caused by neurotransmitter chemicals can be either depolarizing, often but not always resulting in an excitatory postsynaptic potential, or EPSP, or hyperpolarizing, resulting in an inhibitory postsynaptic potential, or IPSP. Postsynaptic potentials generally move passively along the dendritic membrane, gradually becoming smaller as they spread. Therefore, the postsynaptic potentials from more distant synapses will decay more than PSPs from synapses closer to the integration zone at the axon hillock. The postsynaptic potentials produced at most synapses are usually well below the threshold for generating postsynaptic action potentials. How then can synapses transmit information if their postsynaptic potentials are subthreshold? Suppose that two excitatory endings are activated, causing local depolarizations of the cell body. Taken alone, neither would be sufficient to trigger an action potential, but when combined, the two depolarizations sum to depolarize the membrane in the hillock region to threshold. When inhibitory synapses are active, the membrane potential tends to be stabilized below threshold because they induce hyperpolarizations or subthreshold depolarizations that cannot reach threshold. These postsynaptic effects also spread passively, dissipating as they travel. Because some potentials excite and others inhibit the hillock, these effects partially cancel out each other. Thus, the net effect is the difference between the two. The neuron subtracts the IPSPs from the EPSPs. Postsynaptic effects that are not absolutely simultaneous can also be summed because the postsynaptic potentials last a few milliseconds before fading away. The closer they are in time, the greater is the overlap and the more complete is the summation, which in this case is called temporal summation. The summation of potentials originating from different physical locations across the cell body is called spatial summation. Only if the overall sum of all the potentials, both the EPSPs and IPSPs, is sufficient to depolarize the cell to threshold of the axon hillock is an action potential triggered.
Welcome to another episode of Interactive Biology TV, where we're making biology fun. My name is Leslie Samuel, and in this episode, episode 19, I'm going to be answering the question, what is summation? And I'm going to talk about the two types of summation. So let's get right into it. Summation is basically the addition of postsynaptic potential. To illustrate this, I'm going to draw our membrane potential graph with membrane potential on the y-axis, E sub m, and time on the x-axis. And I'm going to start with our resting membrane potential somewhere around negative 70 millivolts. So let's say this is negative 70. Now we've spoken about threshold, and we said that in order for there to be an action potential, the membrane potential needs to reach that threshold, and I'm going to say that's around negative 55 millivolts. So an action potential comes down an axon. It reaches the emitters are released, and that causes in the membrane potential. Now we know that the threshold is up here, so that bump is not going to be enough to cause an action potential. So what needs to happen is we get an excitatory postsynaptic potential, and before the first one finishes, another one comes along, raising the membrane potential even more, and that process continues over and over until eventually we have enough stimulation to cause the membrane potential to reach threshold, and then we get the action potential with our depolarization, our repolarization, our hyperpolarization, and then the action potential is finished. So that's what we're talking about with summation. We're basically adding these postsynaptic potentials. Remember, sometimes we can get excitatory postsynaptic potentials. We can get inhibitory postsynaptic potentials that bring our membrane potential even farther away from the threshold. But we're basically adding them so that we can reach that negative 55 millivolt threshold and cause an action potential. With that understanding, let's look at the two types of summation. We have temporal summation and we have spatial summation. And I'm going to do a drawing to illustrate both. With temporal summation, what we have is a presynaptic neuron. So let's just draw an axon, and I'm going to simplify it by making it just one axon terminal. So here's my neuron. Here we have the releases neurotransmitter, and that causes an excitatory postsynaptic potential in this cell. And that is where we see the first bump. Now, if it sends that signal and it sends another one quickly and another one quickly and another one quickly, and it continues doing that, that's going to cause the membrane potential to go up and up until it reaches the threshold and causes the action potential. This is temporal summation. Now that's different from spatial summation. With spatial summation, we have an axon here, and I'm just going to draw one axon with one. I'm going to draw one axon with Weaver. We also have another axon that comes, and we have an axon terminal that connects to the same neuron. And what's going to happen here is this one can cause a signal, cause an excitatory postsynaptic potential, and before that dies off, we can have another signal coming from this other cell. So we have two separate neurons causing responses in this cell, and of course we get the same result where the membrane potential goes up, and when the next one fires, it goes up again, and this one can fire again, that causes it to go up, and then it eventually reaches the threshold, causing our action potential. That is spatial summation. So an easy way for you to remember this, at least this is what works for me, spatial summation is separated by space, okay, because we have two separate neurons that are firing. Temporal summation is the same neuron firing, but each signal is separated by time. So temporal summation separated by time, same neuron. Spatial summation is separated by space because there are two separate neurons. That's all the content for this video. We've looked at the two types of summation, and we've answered the question, what is summation? If you have any questions, go ahead and leave.